I've known Jeff for many, many years. We're old friends. But, Jeff is one of the old guard of nucleic acid vaccines. He has over 30 years of industrial experience. Started his career at Merck, then moved to Chiron, then Novartis Vaccines, and then finished off the last 5 years of his career at GSK. So he's worked on DNA vaccines, viral replicon particles, self-amplifying RNA. He's unusual, having spent 30 years in industry, having published over 200 peer-reviewed publications. At least 20 of those I co-authored with him in our time from 2008 to 2015 at Novartis Vaccines on self-amplifying RNA. Pleasure to have you here, Jeff, and look forward to your talk.
Well, thank you so much, Andy, for the kind introduction. It's my honor and pleasure to be part of the symposium. I think it's safe to say that vaccines are one of our most effective of medical interventions, and the spectacular success that we've had over the last year with COVID and the speed with which we've come up with and implemented a solution is a beautiful example of the power and the impact that vaccines can have on human health. Having said that, though, several major challenges remain for vaccines R&D, including improving existing suboptimal vaccines like the tuberculosis vaccine, which I believe has been the most widely administered vaccine of all time, yet really only is effective for a small slice of the population. And the influenza vaccine, which can be quite effective if there's a good match between the circulating strain and the vaccine strain, but uses antiquated technology in its production and requires reformulation every year. We have major unmet medical needs for which vaccines could play an important role, but have not yet been developed. And a few examples here: HIV, RSV, and cancer. We've been, until now at least, woefully inadequate to responding quickly to emerging threats. And we've seen this with previous outbreaks like SARS-CoV-1, Ebola, MERS, Zika, various pandemic influenzas, and clearly there's room for improvement, and we really need to throw out the rule book on how we respond. And then finally, the vaccines R&D process itself is quite lengthy and complicated and costly, and there's definitely room for improvement in streamlining how we develop and discover vaccines. To understand what needs to be done and how we might achieve it, I think it's instructive to step back and look at what we currently have in our arsenal of vaccine technologies. The most effective of these vaccines types are based on live organisms typically. Because they launch a live infection, they induce potent and broad immunity and training the immune system as if it were responding to a disease infection. As a consequence, we elicit typically both antibodies and cellular immune responses, including cytotoxic T lymphocytes. But because they're based on live pathogens, although engineered to be safe, there's always the potential safety risk of reversion to a virulent state. And it's not always feasible to develop this type of vaccines. As a consequence, the timelines for R&D of these kinds of vaccines can be quite lengthy. To increase the safety profile of vaccines based on whole organisms, we move to inactivate them, typically through chemical means. And this obviously makes them safe because we eliminate the risk of infection. And because using this approach, we don't need to attenuate them, you have a simpler R&D process typically. However, because they're not establishing an infection, you have more narrow immunity, typically limited to protective antibody responses. The chemical inactivation can sometimes have a detrimental effect on the integrity of the antigen. And like live organism-based vaccines, it's not always feasible because not all organisms are amenable to growth in cell culture. And also, because they have all of the antigens of the pathogen, sometimes they have antigens that are not relevant or even detrimental to immunity. So to address that, we move to subunit-based vaccines where we purify or express only the key elements that we want to target the immune response against, be it a recombinant protein or a polysaccharide. This retains the safety profile of inactivated organisms, but with a more defined composition. But like inactivated organisms, you typically induce only antibody responses, or at least immunity based on antibody responses. They often require adjuvants or carrier molecules to increase potency, and they're complicated to make. And this set the stage for the impetus for nucleic acid vaccines to combine the positive attributes of these various technologies, but avoiding the limitations. And by that, I mean to induce the broad and potent immunity, including both antibodies and cellular immune responses, but with the safety and simplicity of a subunit vaccine approach. The various types of nucleic acid vaccines are listed here, and I've included live attenuated viruses because by definition they contain the genome of these organisms, but most commonly it's either viral vectors or plasmid DNA or mRNA. And I'm going to focus my attention here really on the simplest two of these nucleic acids, the plasmid DNA and the messenger RNA. Plasmid DNA typically is expressed in E. coli as a bacterial plasmid through fermentation, although technologies are being developed which will enable us to make plasmid DNA vaccines completely synthetically in the absence of fermentation. But the vaccines that have been used up to date look like this cartoon here. They contain a promoter, a gene encoding the antigen, and a terminator, which our cells use to transcribe the mRNA and express the protein off of the mRNA. And for those plasmids that require bacterial fermentation, there's also a bacterial selection gene that's used. The broad utility of this approach has been well established in literally thousands of scientific papers that have been published since the early 1990s. And there have been several hundred clinical trials involving thousands of subjects demonstrating the safety of this approach and in some cases immunogenicity against the antigens exposed. And importantly, we have several licensed products now for animal health applications that proves the principle that the DNA vaccine technology can be used, can be effective, and is even in large animals like horses and cattle. mRNA vaccines, on the other hand, are shown here, and they come in two basic flavors, so-called conventional mRNA vaccines, because they look very much like the mRNAs that are found in all of our cells, including a 5' cap, 5' untranslated region, the sequence of the gene that you're encoding, a 3' untranslated region, and a poly A tail, all of which are important for effective expression in eukaryotic cells. They come in two basic formats, one that includes natural bases, that is the bases that are found in all of the mRNAs in our cells. And this is the approach that companies like CureVac are taking in producing their mRNA vaccines. But you can also use modified bases, most commonly pseudouridine can be incorporated instead of uridine. And this is the approach that BioNTech, Pfizer, and Moderna are using, with the potential advantage here being that it may make them more immunologically silent to the innate immune system and possibly confer better stability onto the molecule. The other type of mRNA is so-called self-amplifying mRNA, or SAM for short. It has the same basic elements of the cap, the UTRs, the antigen gene, and the poly A tail, but differs fundamentally in that it's based on an engineered viral genome, RNA virus genome, where in addition to the antigen gene, you encode the genes of the replicase machinery that when get into a cell, it uses the cell translation machinery to express these non-structural proteins, NSP1 through 4, which together form the RNA polymerase, which makes many copies of the incoming RNA, and thereby possibly allowing us to use lower doses of RNA to achieve the same effectiveness as a conventional RNA. These also come in two formats. Most commonly, the replicase and the antigen genes are encoded on the same mRNA molecule, with the antigen gene being driven by a subgenomic promoter. But two examples of separating the replicase genes from the antigen genes have been published, one by BioNTech and one by Robin Shattuck at Imperial College. There's also a large literature, albeit not as large as DNA at this point, but certainly growing fast, showing the broad utility of this approach in animal models. And there have been a limited number of human clinical trials of various kinds so far, but obviously the breakthrough was the remarkable efficacy that we saw with 2 of these mRNA vaccines based on the conventional mRNA vaccines with the modified basis of Moderna and BioNTech/Pfizer, leading to the emergency use authorization for COVID and the success that we've had so far. Now, it may seem like this happened quite suddenly, but in fact, it has been quite a journey for nucleic acid vaccines dating back more than 3 decades now to the seminal paper that was published by John Wolfe in collaboration with Vical, showing that simple injection of either a pure plasmid DNA or mRNA could result in expression of the protein in the cells of the immunized animal. And this set the stage for application for a variety of purposes. But when we first learned of this observation, we immediately thought, well, this could be a great way to not only induce antibodies against the expressed protein, but because you're mimicking a virus infection in some ways, you could also possibly induce CTLs this way. So this really set in motion a lot of effort, both in industry as well as academic and government laboratories. It was only a few years later that the proof of principle that an expressed protein this way could induce an antibody response, and that was published by Stephen Johnson's group using the gene gun to deliver a plasmid DNA on gold beads. The following year, we published that CTLs could be induced as well, and showed that these CTLs and antibodies could confer protective immunity in an animal model of influenza. At the time, we also compared mRNA, and we did a direct comparison of mRNA with plasmid DNA encoding influenza nucleoprotein, and showed that both of these approaches worked equally well in inducing antibodies and CTL. But at the time, we didn't see a path forward to industrializing the mRNA approach because of the relative instability of the mRNA molecule and the perceived cost that it might take to manufacture an RNA product. So we and most of the field really at that point on focused on plasmid DNA. However, there was a paper published by Pierre Mollien's group in '93 as well, showing that CTLs could be induced by mRNA encoding the nucleoprotein. And then that was followed a couple of years later by work by Conry et al. Showing that mRNA could induce potent immune responses against cancer antigens. Because of the growing interest in the technology, DNA in particular, the FDA and the WHO both educated themselves about the technology and published guidance documents for industry on points to consider for manufacturing of DNA-based products. And I think that was really an important step forward for the field to allow people to really know the path, or at least have a blueprint for the pathway for how you might take a DNA vaccine or an RNA vaccine forward to clinical trials and eventually licensure. The first clinical trials of DNA occurred beginning in 1996 or so, with work in HIV-positive individuals by David Weiner at the University of Pennsylvania and Britta Warren at the Karolinska Institute. And at that time, we also started a trial for an influenza DNA vaccine back at Merck. As I mentioned, we have licensed products for animal health, and they began to be licensed in 2005. We have 2 licensed vaccines for farmed salmon. We have one for West Nile virus in horses. We have one for a melanoma vaccine for dogs. And then we also have a DNA product that's given to swine to increase their body weight. So as I said, there's really a strong proof of principle that at least in some indications and even in large animals, DNA vaccines can be effective. Over this period of time, a large number of clinical trials were undertaken for DNA vaccines. most of which gave modest immune responses, but there were several examples of good induction of functional immune responses by DNA vaccines, including one against the West Nile virus, once against the SARS-CoV-1, one against Ebola, and one against Zika, all of which indicated that even in humans, potentially protective immune responses could be achieved, at least for certain indications. For mRNA clinical trials, the first trial began in 2007 by researchers at Tübingen University in collaboration with CureVac for cancer indications. Andy and his team at Novartis published first on the synthetic versions of self-amplifying mRNA showing antibody induction, CTL, and protective efficacy in 2012. And all of this has laid the groundwork for the remarkable results that we saw with the mRNA vaccines for COVID last year. And this evolution of initial meteoric rise for DNA vaccines, followed by a more gradual rise of mRNA vaccines, is reflected here in how the scientific publications that have appeared over this time on DNA in blue and RNA in red, showing that there's been a steady rise in the interest in RNA vaccines over time, with a spike, if you'll permit me the pun there, for the last year for mRNA, owing largely to the huge increase in the interest and application of RNA vaccines for COVID. So if we go back to the challenges, I'd like to walk through some data that I believe suggest that nucleic acid vaccines and RNA in particular could play an important role in addressing each and every one of these challenges. With regard to new and improved vaccines, clearly we've seen the success with COVID, and time will tell how broadly applicable that is, but we do at this point have a impressive and comprehensive preclinical package of data using RNA-based vaccines in animal models. And this table here summarizes the work that we did at Novartis and GSK using the self-amplifying RNA in a wide variety of different disease models, mostly viral targets, but also other targets like bacteria, GPS, GAS, as well as malaria, and you'll hear a lot more about that from Rick Bucala shortly. But you can see that in these models, we were able to induce immune responses, and where possible to test, we proved efficacy in a pathogen challenge model as well, including in several non-human primate models, like for CMV, rabies, influenza, HIV, and Zika. And importantly, where possible to benchmark with licensed human vaccines, such as for rabies and flu, we demonstrated that the SAM technology was as good as, and in some cases better than, the full human dose of the licensed product that's known to be effective in humans, which bodes well for the technology in humans. In addition, a recent review paper published by Andy and his colleagues listed several other targets that others have used the self-amplifying RNA for, including several human clinical trials that have been conducted and are ongoing, such as the GSK trial for rabies, various trials for SARS-CoV-2, and the personal neoantigen cancer vaccine approach that we'll hear from Karen about later on from Gritstone. For the response to emerging threats, as I said, we have been woefully inadequate up to now. But the success that we've seen with COVID, I think, validates the technology for the speed with which we can bring to bear on these types of situations. Looking back to 2013, this was our first opportunity to really test this hypothesis of speed. And really, it was a real-world live fire exercise, because this was related to the 2013 H7N9 outbreak of influenza. Because of an ongoing relationship we had at the time with the J. Craig Venter Institute, we were able to quickly synthesize the gene encoding the hemagglutinin molecule based on the sequence that was published on the internet back in 2013 on Easter Sunday. Within about a week, we had RNA that was ready to use for vaccines that was shown to be pure, expressed the HA molecule, and then when immunized into mice, induced potent neutralizing antibody responses within a couple of weeks after a single injection, indicating that, albeit this research exercise, from a technical perspective, one can respond very quickly from the sequence to have material ready for deployment. And of course, this being a research exercise doesn't reflect very accurately what might happen to try to bring a vaccine to human clinical trials. But nevertheless, there are two recent examples of how quickly you can go from pathogen identification, antigen identification optimization, to manufacturing, release, and administration of these vaccines in humans in phase 1 clinical trials. The first being the Inovio response to Zika with the pre-M and E antigen sequence to phase 1 trials in about 4 months. And then, of course, the remarkable speed with which Moderna was able to go from sequence to start of clinical trials in just a couple of months. So a vision for a global response platform could be something like this, that when the next outbreak is declared, the pathogen identified and sequenced, that information could very instantaneously be transferred to the laboratories where the vaccine would be made. The early work could be done in silico, and for example, with a pandemic influenza outbreak, one could very quickly focus in on the hemagglutinin molecule and synthesize that immediately. But for something like a pathogen that we have little experience with or information about, that would obviously take some discovery work to understand what is the optimal sequence to put in the vaccine. But as soon as we have that, then just in a matter of weeks, we could have vaccine ready to deploy. One of the formulations that we were developing at Novartis and GSK was based on a cationic emulsion. And this adds to the potential utility of a rapid response because that could be stockpiled separately from the RNA. You could have a stockpile of GMP ready to go formulation and then simply respond by making the RNA and then mixing it with the formulation. And then finally, the process to make vaccines is long and complicated and costly. And there's certainly room for improvement in streamlining this process. Typically, it can take decades or more to go from the discovery phase to the registration phase. And as a consequence, if we look back to many of the vaccines that are in wide use today, it has taken a very long time from their discovery to having a vaccine ready to go. And while we're getting better with time through the introduction of technologies, we're still takes a long time to develop a vaccine. And albeit for targets like HIV, this is due to reasons of complications of the disease itself. But in many cases, a significant contributing factor to the length of time it takes is because of the fact that these vaccines that we use today are biologics. And by that, I mean that we use biological systems like cell culture to produce them. And in contrast, nucleic acid vaccines and RNA in particular are completely synthetic. In the absence, we've removed the requirement for cell culture out of the equation altogether. And RNA drug substance can be produced from the DNA template very quickly through a transcription reaction, as we all know, and a couple of purification steps to, in a matter of hours, having highly pure RNA drug substance that's produced consistently in just a matter of hours. And importantly, because the RNA itself from one product to the other differs only by virtue of the nucleotide sequence of the gene insert, from a physicochemical perspective, the products are very similar. And this allows us to potentially have a production process and purification process that is generic. And this will be very important in establishing standardized methods to produce and purify the product, going from one product to the next, whereas with biologics, this has to be done really in a product-specific manner for every new product, and it takes a long time. Another rate-limiting step is the characterization and release of products. For biologics like recombinant proteins, most of these release tests need to be developed de novo for every new product and can take several years from the time you start to have validated assays for releasing your product. In contrast, the mRNA platform, and in particular the self-amplifying mRNA platform, most, if not all, of these assays could be generic and, once established, used for every new product going forward. So that together, this could result in a dramatic reduction in the amount of time it takes to discover and develop a vaccine, thereby getting the vaccine to people who need them much faster. And I've shown here by the yellow stars, the opportunities for acceleration that could occur by using the nucleic acid vaccine technology. And undoubtedly, our successes for COVID in the past year have been the product of a massive concerted effort by industry, both pharmaceutical and biotech industry, working in partnership with foundations, government labs, the regulatory agencies, including the massive investment that was made that has allowed us to take certain parallel activities at risk that you wouldn't normally do. But I think it's safe to say that the preceding 3 decades of work on nucleic acid vaccines, and in particular DNA vaccines, has given us a solid foundational knowledge on which we've drawn upon for application to mRNA. For example, mechanistically, because RNA and DNA conceptually function the same way, the understanding that we had on where the antigens are expressed, for how long, the mode of priming of antibodies and T-cell responses, and the role of innate immune stimulation in this helped us to hone in on the right questions to ask as it came to RNA vaccines. From a safety perspective, 2 of the initial concerns, because DNA vaccines would result in low levels of antigen expression over a period of time, primarily in non-antigen presenting cells, would this result in immunologic tolerance or conversely autoimmunity? And the data safety package we have with DNA, I think, gave a level of comfort that this was not likely to occur for RNA as well. The breadth of utility, not only in the animal models, but also the clinical trials, the success with animal health products, and the fact that these nucleic acid technologies can be used to facilitate screening in the discovery phase, I think allowed us to take a much more streamlined approach when it came to applying mRNA. And then from a practical perspective, in a technology readiness perspective, the comfort level and the familiarity that regulatory agencies had with nucleic acid products during the DNA years has allowed us, I think, to move much more quickly in embracing the mRNA technology. And then logistically, because of the potential of DNA in the early days, that it would be a game-changing technology, it spawned a legion of supporting industries, CROs, CMOs for manufacturing raw materials, product, reagents, and assessment of these products. And this, I think, allowed us to move much more quickly in response to the massive need that we had suddenly with mRNA. So today we have a toolbox of technologies consisting of both plasmid DNA and mRNA. And while they have common attributes in that they can be made quickly, they have generic processes to manufacture, purify, and characterize and release the products, enabling both a rapid response as well as individualized vaccines. They have unique attributes and limitations that in some ways make them complementary. For example, plasmid DNA is thermostable, even at ambient temperatures, can be manufactured at large scale and at low cost, and therefore its feasibility for global use, particularly in the developing world, I think it is quite attractive. mRNA, because it requires only delivery into the cytoplasm, I think lessens the dependence on delivery compared to plasmid DNA, and likely is a contributing factor to their increased potency. And obviously the proof of principle that we've had for COVID really will set the stage for a lot of further interest and investment in the technology. On the limitation side, with DNA, obviously, so far the potency in humans has been limited to just a few examples of certain targets where we can induce potent functional immune responses. This may be more a aspect of the antigens themselves being relatively inherently antigenic and immunogenic. But I think there's still opportunities for us to better understand what is really the species barrier here for plasmid DNA vaccines, because if we could break that down even a little bit, it might open up the possibility of using plasmid DNA, particularly for developing world applications. On the mRNA side, we know that mRNA is more inherently immunostimulatory than DNA, and that likely plays a role in whether the vaccines work or not. However, there's the potential for these immunostimulatory effects to be detrimental as well, and we know that if you overstimulate pro-inflammatory immune responses too early, that can prevent launch of the RNA vaccines and therefore limit their effectiveness, but also they may contribute to tolerability issues if the right balance is not struck between safety and effectiveness. So I think we still have a ways to go to understand mechanistically what is happening here so that we achieve this right balance. So in summary, I believe over the past 3 decades, we've seen that nucleic acid vaccines have great potential, not just in the discovery phase to facilitate screening of antigens, and it's certainly, even if in the end you choose to make a recombinant protein-based vaccine, you can streamline discovery by testing your different forms of the antigen in the context of a nucleic acid, which is much easier to make than a recombinant protein. Because the technology mimics a live virus infection, it has the potential to hit that sweet spot of inducing broad and potent immune responses, but in a safer and simpler manner than live attenuated vaccines. Because of the plug-and-play fashion of the way that the vaccines are manufactured, there's a real possibility here that we can streamline the R&D process overall, and thereby reducing the time it takes to get effective vaccines to people who need them quickly, but also, having a much more effective rapid response to newly emerging pathogens. And I believe that the vaccines applications that we've seen here and the successes for COVID is certainly going to spur a lot more interest and investment, not just for infectious diseases against other targets, but also application of the technology for other non-infectious disease indications like gene and cell therapy, gene editing, oncology. And I believe that the next 5 years is going to be quite an interesting one. It remains to be seen how broadly effective the technology is, but we're going to give it a good shot. I will stop there and be happy to answer any questions. Thank you for your attention.
Great. Thank you very much, Jeffrey. That was fantastic. It was a really great talk. I really appreciate you walking us through really kind of some of the history of how we got to this point and what we learned from DNA vaccines to get here.